So I'll be talking about the pentose phosphate pathway and its role in diabetes and cancer. So first I'll be talking about the pentose phosphate pathway, which we learned about a couple weeks ago in chapter 14. So the pentose phosphate pathway, or the PPP, is an alternative pathway of glucose metabolism. The pentose phosphate pathway starts after splitting off from the first step of glycolysis, where it produces fructose 6-phosphate and glyceraldehyde 3-phosphate from the intermediate glucose 6-phosphate. This pathway takes place mostly in the cytosol of liver and adipose tissue cells. NADPH and ribose 5-phosphate is generated from this pathway. Both of these products are useful for cell survival and proliferation. It is also important to remember that ATP is not produced in this pathway. So continuing on the pentose phosphate pathway, it has an oxidative phase and a non-oxidative phase. This oxidative phase will generate NADPH for biosynthesis of fatty acids, sterols, nucleotides, and non-essential amino acids. NADPH is the reducing power required for these reactions. The NADPH-derived conversion of oxidized glutathione to reduced glutathione with the help of the enzyme glutathione reductase is very important for antioxidant defenses. It also helps produce reactive oxygen species by acting as a substrate of NADPH oxidases. Ribose 5-phosphate is the other product generated during the oxidative phase of the PPP. RP5 is the building block for nucleic acid synthesis. So the oxidative phase of the PPP. Glucose 6-phosphate dehydrogenase is a rate-limiting step in this phase. It controls the flow of glucose 6-phosphate into the pathway. It also catalyzes the conversion of GP6 to 6-phosphoglucosamine lactone, producing NADPH. And you can see how it goes on further down the line in this diagram from the textbook in chapter 14. So more on the oxidative phase of the PPP. The oxidative phase is the largest contributor to the cytosolic NADPH in mammalian cells, and it has other contributing enzymes like malic enzyme 1. You can see there's 2 more, and then there's also mitochondrial NADPH production, which involves the 5 key enzymes listed below. So now on the non-oxidative phase of the PPP, there are a series of reversible reactions that involve the transfer of chemical groups. Some are ribose 5-phosphate isomerase and ribose 5-phosphate epimerase catalyze reversible reactions that convert ribose 5-phosphate into ribose 5-phosphate and X5P. And these next couple of reactions I'll show in better detail in a diagram on the next slide in a second. But TKs will move 2 carbons, and TALDOs will move 3 carbons, which you can see on the next slides that I got from the chapter 14 lecture slides. So the main purpose of this phase is to regulate the flux of glycolysis by providing fructose 6-phosphate and glyceraldehyde 3-phosphate. It also restores metabolites from the oxidative phase. So here's where I'm talking about, where you can see TALDOs can help with the conversion of S7P and G3P to E4P in fructose 6-phosphate. And then here's the other reaction where you can see TKs will convert X5P and E4P to G3P and fructose 6 phosphate. And they're both reversible reactions. So this is a diagram that I got from the paper, and you can see here that this is where glycolysis starts, and then after the first step, it branches up. The PPP starts because it branches off from the first step, and you can see follow down the pathway to where it has its oxidative phase and its non-oxidative phase, and all of the enzymes for oxidative and non-oxidative phases are highlighted in green here. So now we'll get into the part where it talks about type 2 diabetes and how the pentose phosphate pathway plays a part in it. So type 2 diabetes is a chronic illness that is characterized by persistent hypoglycemia, and it has two main features, which are insulin resistance and increased insulin secretion. So insulin resistance can be characterized by decreased glucose uptake by liver, adipose tissue, and skeletal muscle. And then in increased insulin secretion, you can see that pancreatic beta cells are compensated, which lead to hyperinsulinemia and beta cell dysfunction. Main complications of type 2 diabetes are eye damage, liver damage, and kidney damage. So the main takeaway about obesity-related insulin resistance I want to talk about is that the chronic inflammation due to obesity is a key contributor to insulin resistance. And you can see these points talk about how some of the inflammation that can lead to insulin resistance, like in the skeletal muscle and liver, which creates systematic insulin resistance, and elevated levels of circulating free fatty acids impair insulin action, increase hepatic glucose production, and may lead to conditions like non-alcoholic steatohepatitis. Here's where you see how the pentose phosphate pathway is related to obesity-related insulin resistance, because you can see like here excess NADPH can lead to oxidative stress in the liver, which will worsen insulin resistance, and then CKLP will regulate the flux through the PPP and is important for macrophage polarization, and also increased G6PD expression in ATMs and adipocytes contributes to inflammation and insulin resistance. Here is a figure from the paper where it shows information from the last slide in a diagram with the CARKLs, the M2 macrophages, M1 macrophages, and adipocytes, and it shows increase and decrease of G6PD and what it will do for inflammation and insulin resistance. So another way that PPP is involved in insulin secretion now, you can see that NADPH is a key modulator in the amplifying pathway as it converts oxidized glutathione to reduce glutathione, facilitating insulin granule exocytosis via the enzyme SYNAP1, which you will see more on the next slide in a figure. So more on the role of PPP in insulin secretion. The optimal activity of G6PD and 6PGD enzymes that produce NADPH is necessary for effective GSIS. And Really, the PPP, NAD, PH, GSH, and SNAP1 pathway must be precisely regulated to support effective GSIS and mitigate the effects of diabetes. So this figure is also from the paper, and you can see here that NADPH from the pentose phosphate pathway converts oxidized glutathione to reduced glutathione right here, and GSH elicits insulin granule exocytosis via SNAP1. However, NADPH might inhibit insulin secretion by promoting NADPH oxidases, which is this right here. So I found this diagram in the textbook in section 14.6, and it shows a pathway of how NADPH and glutathione protects the cells from ROS. So for PPP and its role in chronic diabetic complications, you can see that there are several complications that diabetes can lead to, and really the main thing is that thiamine deficiency and reduced TKT activity are linked to diabetic complications. So now going into how PPP plays a part in cancer. So NADPH and R5P help regulate DNA damage response, metabolism, and proliferation in cancer cells. Many enzymes of the PPP have shown to play a part in metabolism and regulation of cellular activities. And this little diagram was also in the paper, and it is showing the regulation of G6PD in cancers. As you can see, there are several different signaling pathways that contribute to this. Now we'll talk about oxidative enzymes in the PPP. They increased levels of G6PD are commonly observed in various cancers driven by multiple signaling pathways. And you can see that there are multiple key regulators, which I'm not going to read them all, but you can read for yourself how it will affect everything. So the main one I want to talk about on this slide is that suppressing G6PD can induce cellular senescence and increase sensitivity to chemotherapy and hepatocellular carcinoma. And the increased G6PD enhances the oxidative pentophosphate pathway, providing ribose 5-phosphates for nucleotide synthesis and NADPH for redox balance. So there are a couple of things that non-oxidative enzymes in the PPP can affect in cancer cells, like elevated 6-PGD levels are associated with shorter survival in breast cancer and are implicated in various cancers, including liver and thyroid, breast, and others. You can see more on how they can affect cancer cells. So continuing of the non-oxidative enzymes in PPP, TK's expression is elevated in various cancers and is regulated by factors like BACH1, KEAP1, and NRF2. TKT enhances in non-oxidative PPP, contributing to chemotherapy resistance in leukemia and pancreatic cancer. Serum fructose levels in pancreatic cancer can induce TKT expression, promoting nucleic acid synthesis. TKT maintains redox homeostasis in liver cancer cells, and its inhibition leads to increased ROS and decreased glycolytic flux. And surprisingly, it also supports tumor growth through mechanisms independent of its enzymatic activity. Lastly, here are my 2 questions for y'all.